had this really profound realization today.
Oh, yeah. What happened?
Well, it completely shattered a mental image I've held since I first looked through a microscope in middle school.
Let me guess. The classic biology class visual.
Yes. You know how we're typically taught to visualize a cell? We picture this static, microscopic blob of jelly.
Right, a tiny little inert dot on a slide.
Exactly, just a dot. Well, going through the materials for this deep dive, it hit me like a freight train. That blob is a total lie.
It really is.
If you actually scale it up, a cell is this hyper-dense, bustling, fiercely organized metropolis. We are talking about highly secure border walls with active customs agents, sprawling manufacturing districts, complex waste management facilities.
A central command vault.
Yes, and independent power contractors running the grid. It's an entire universe of physics and chemistry functioning flawlessly inside a microscopic drop of fluid.
I absolutely love that you're bringing that exact energy to this session. Welcome, everyone, to today's deep dive.
We are so excited for this one.
We really are. We are taking a very specific, highly structured look at the fundamental unit of life. We're adopting what the scientific literature calls a physicochemical approach to life.
Which is the core of reductionist biology, right?
Exactly. Think about the most staggering mysteries of human physiology, like how your immune system orchestrates a targeted attack against a novel pathogen.
Or how the synapses in your brain are physically rewiring themselves to form memories of the words we're speaking right now.
Precisely. To truly grasp those gross physiological and behavioral phenomena, you cannot just look at the whole organism.
It's too complex.
Way too complex. You must reduce the system down. You have to analyze the living tissues for their elements and compounds and ask, what are these molecular structures actually doing inside this metropolis?
So we are stripping away the macro. We're diving into the molecular basis of life.
We are.
Taking the cold, hard rules of physics and chemistry and seeing how biology exploits them. But before we zoom all the way in to map the streets of this cellular city, I want to ask you the big existential question.
Lay it on me.
Let's cut straight to the essence. Why does this structural topic even exist? I mean, what ultimate problem does the cell solve in the natural world?
That is the perfect place to start. The essence of the cell boils down to fighting entropy through compartmentalization.
Fighting entropy. So fighting chaos.
Yes. Life, fundamentally, requires a very specific, highly regulated chemical order to exist. And that delicate order must be aggressively protected from the chaotic, random physics of the outside inanimate world.
You need a wall.
You need integrity. The core idea here is the absolute requirement of integrity. Unicellular organisms are defined by their capability to do two things. One, maintain an independent existence.
Okay.
And two, perform the essential functions of life. Here's the critical takeaway for you listening. Anything less than a complete structure of a cell does not ensure independent living.
Wait, so you could have all the right ingredients?
You can put all the right proteins, the right nucleic acids, the right lipids into a test tube. You can mix them up.
But it's not alive.
It's not independent life. If they are not structured, bounded, and compartmentalized into a complete cellular architecture, you do not have life. The cell is the fundamental structural and functional unit that provides that vital integrity.
It is the definitive boundary line between the living and the non-living.
Exactly.
So without the city walls, the complex economy inside just dissolves into the surrounding wilderness.
It just diffuses away.
Wow. And speaking of understanding that intricate architecture, our sources provided this really fascinating historical prologue about G. N. Ramachandran.
Ah, yes, the founder of the Madras School of Conformational Analysis of Biopolymers.
That's the one. What struck me was that he didn't even start in biology. He was heavily influenced by his mathematician father.
Right.
So how does pure mathematics unlock the secrets of structural biology?
It is a brilliant example of how cross-disciplinary thinking reshapes our understanding of the world. While Ramachandran was at Cambridge, he crossed paths with Linus Pauling.
Linus Pauling.
The very same. He was deeply inspired by Pauling's published models of the alpha helix and beta sheet structures. That mathematical and physical inspiration directed Ramachandran to tackle the structure of collagen.
Which is everywhere in our bodies.
Exactly. By 1954, he published the triple helical structure of collagen in the journal Nature. He then went on to analyze the allowed conformations of proteins through a mathematical framework.
And that framework is the famous Ramachandran plot, right?
Yes. Universally known as the Ramachandran plot. His legacy proves that massive macro-level discoveries in medicine and biology almost always originate from a rigorous mathematical understanding of basic molecular geometry.
Form dictates function.
Always.
It sets the perfect tone for what we are doing today. So, holding on to that structural mindset, I want to build a mental map for you, our listener.
Let's do it.
We are going to zoom in layer by layer into this biological metropolis. But before we start inspecting the individual factories and power plants, we need to know who actually drew the map.
Who figured it all out?
Right. Who figured out that the entire biological world is constructed from these individual units.
That brings us to the formulation of the cell theory, and we need to look at the big three historical figures here.
Okay, who's first?
We start in 1838 with Matthias Schleiden. He was a German botanist. He examined a massive, diverse array of plants and made the sweeping observation that all plants are composed of different kinds of cells.
And those cells organize together to form the tissues of the plant.
Exactly.
So Schleiden covers the botanical side. Then, literally the next year in 1839, Theodor Schwann enters the picture.
Yes.
Now, Schwann was a British zoologist. He was studying different types of animal cells and reported that these animal cells had this incredibly thin outer layer,
Which we now recognize as the plasma membrane.
Right.
Now, let's pause right here. For anyone listening who is preparing for a high-stakes exam like Neet, this next point is a classic trap that examiners use year after year.
Oh, lay it out for them.
Schwann was a British zoologist. His primary mandate was animal cells. However, based on his comparative studies involving plant tissues, Schwann is the one who concluded that the presence of a cell wall is a unique character of plant cells.
Wait, I see the trap. The zoologist is the one who defined the defining feature of the plant cell.
Yes.
That feels like a trick question waiting to happen.
It absolutely is. They will present a multiple-choice question asking which scientist concluded that the cell wall is unique to plants.
And the instinct for a rushed student is to pick Schleiden because he's the botanist.
Exactly. You must remember it was Schwann, the zoologist, who made that specific conclusion regarding the cell wall. And on the basis of that holistic view, Schwann proposed the hypothesis that the bodies of both animals and plants are composed of cells and products of cells.
So together, Schleiden and Schwann officially formulated the cell theory.
They did.
But looking at their original theory, there's a glaring plot hole. They brilliantly deduced that everything living is made of these building blocks, but they couldn't explain the origin of the blocks themselves.
They hit a wall.
Right. How does the city get new buildings?
That gap remained wide open until 1855. That's when Rudolf Virchow stepped in. Virchow revolutionized the theory by explaining that cells divide and new cells are formed exclusively from pre-existing cells.
The famous Latin phrase.
Omnis cellula e cellula. He modified the hypothesis of Schleiden and Schwann to give the cell theory its final, complete shape.
Omnis cellula e cellula. All cells arise from pre-existing cells. If we pull back and think about the implications of that for a second, it's staggering.
It really is.
It means there's an unbroken physical lineage of cellular division stretching from the cells in your body right now, all the way back through evolutionary history to the very first primordial cells billions of years ago.
You are the current manifestation of a continuous, uninterrupted chain of life.
It is profoundly humbling. So to summarize our mental map foundation, the modern cell theory rests on two main tenets.
1, all living organisms are composed of cells and products of cells.
And 2, all cells arise from pre-existing cells.
Spot on.
With the map drawn, let's talk about the incredible diversity of these metropolises. Because once you establish that everything is made of cells, the sheer variation in how those cells are built is mind-blowing. Let's look at the scale. We know cells are small, but the range of small is vast.
The variations in scale are truly staggering. Let's establish some anchor points for your intuition. On the absolute lowest end of the scale, you have the mycoplasmas.
The smallest of the small.
The smallest known cells, measuring only 0.3 micrometers in length.
Just to anchor that for you listening, a typical bacteria, which we already consider incredibly tiny, ranges from 3 to 5 micrometers. So mycoplasmas are an order of magnitude smaller than regular bacteria.
Yes.
And viruses, which aren't even true cells because they lack that vital independent structural integrity we talked about, sit around 0.02 to 0.2 micrometers.
Tiny fractions.
What about the upper limits of a single cell?
The largest isolated single cell in the world is the egg of an ostrich.
An entire ostrich egg is one cell.
One single cell. And when we look within complex multicellular organisms like humans, Our red blood cells are roughly 7.0 micrometers in diameter.
Okay.
But if we are talking about length, nerve cells are the undisputed giants. Some can stretch over a meter long in large animals.
Form follows function in the most extreme ways. A red blood cell is a biconcave disc, so it can bend, flex, and squeeze through microscopic capillary toll booth while maximizing its surface area to trade oxygen.
Exactly.
A nerve cell is a massive, elongated communication cable. A plant trachea is essentially a hollowed-out pipe for transporting water up a tree trunk. The city architecture perfectly matches its industry.
That principle that biological form strictly dictates physiological function is a lens you should look through for everything we discussed today. Let's apply that lens to the simplest, most rugged outposts in the biological world.
The prokaryotes.
Yes, the prokaryotes.
The prokaryotic minimalists. We are talking about bacteria, blue-green algae, mycoplasma, and PPLO.
Which stands for pleuropneumonia, like organisms?
Right. These are the rapid multipliers. Yeah. No complex organelles, no sprawling bureaucracy, just rapid replication and raw survival.
Exactly. And despite their minimalist internal structure, they exhibit a wide variety of shapes. You need to have the 4 basic architectural shapes of bacteria locked in.
Let's run through them.
There is the bacillus, which is rod-like, the coccus, which is spherical, the vibrio, which is comma-shaped, and the spirillum, which is spiral.
I want to dig into how they survive without all the complex compartments eukaryotes have. Their defining trait is the lack of a well-defined membrane-bound nucleus.
Correct.
The genetic material is just out there naked in the cytoplasm. It's typically a single circular chromosome of genomic DNA, but there's a secondary genetic feature they have that is incredibly relevant to modern medicine.
Ah, you're referring to plasmids. This is a brilliant evolutionary mechanism.
That's so cool.
In addition to the main genomic DNA that runs the basic day-to-day operations of the bacteria, many prokaryotes possess much smaller circular DNA molecules located completely outside the genomic DNA. These are the plasmids.
If the genomic DNA is the factory operating system, the plasmid is like a tiny, downloadable expansion pack that gets passed around between bacteria, granting them special abilities.
That is a highly accurate analogy. And here is what you must remember for exams. Plasmid DNA confers certain unique phenotypic characters to the bacteria.
Like what?
The most famous and clinically terrifying character conferred by plasmids is resistance to antibiotics.
Wow.
When you hear about superbugs in hospitals, you are often hearing about the rapid sharing of resistance plasmids. Furthermore, because they're so easily transferable, plasmids are the foundational tools used by biotechnologists to monitor bacterial transformation when we want to insert foreign DNA into a cell.
That is fascinating. Now, if they're so minimalist on the inside, how do they defend themselves from the outside? Let's look at the prokaryotic cell envelope. It's not just a single wall. It's a deeply integrated security system.
Let's build a clear guide for this.
Yeah.
Most prokaryotic cells, particularly bacteria, have a chemically complex envelope consisting of a tightly bound 3-layered structure.
3 layers.
Even though each layer performs a distinct defensive function, they are fused to act together as a single protective unit.
Imagine a medieval fortress. You have the outer moat, the heavy stone wall, and the inner gate. What serves as the outer moat?
The outermost layer is the Glycocalyx. Its composition and thickness vary wildly depending on the specific bacteria and its environment.
Okay.
In some species, it presents as a loose, viscous sheath known as the slime layer. In others, it is heavily cross-linked, becoming thick and tough. We call that the capsule.
And the capsule is dangerous.
Very. The capsule is particularly notorious for helping pathogenic bacteria hide from a host's immune system.
So the Glycocalyx is the perimeter defense, either a sticky trap or heavy armor. If something gets past that, it hits the second layer, which is the cell wall.
Right.
Since we know a cell is packed with solutes, water is constantly trying to rush inside. Without a heavy structural constraint, the cell would just inflate and pop.
Exactly. The physics of osmotic pressure demand a structural counterforce. The cell wall determines the shape of the cell and provides that strong structural support. Its primary evolutionary job is to prevent the bacterium from bursting or collapsing.
But there's an exception, isn't there?
Yes. Here is a massive exception case you must commit to memory. While practically all prokaryotes rely on this rigid cell wall, there is one notable outlier: mycoplasma. Mycoplasmas completely lack a cell wall.
No cell wall for mycoplasma. That explains why they are the smallest, right? They don't have the structural scaffolding to support a larger size, and they have to live in osmotically stable environments.
Spot on.
And finally, The inner gate of our fortress.
The innermost layer is the plasma membrane. It is selectively permeable, controlling the flow of molecules interacting with the outside world. Structurally, the prokaryotic plasma membrane is remarkably similar to the one we will see in eukaryotes.
Good to know.
By the way, based on the chemical differences in these cell envelopes and how they react to a specific staining procedure developed by Christian Graham, bacteria are broadly classified into two groups.
Gram-positive and gram-negative.
Exactly. Those that take up the Gram stain are gram-positive, and those that do not are gram-negative bacteria.
So we have the security system mapped out. But earlier, we said prokaryotes lack membrane-bound organelles. They don't have mitochondria. So where does the energy come from? How do they replicate DNA without a specialized compartment? If the membrane is just a border wall, how is the city functioning?
This is a high-yield focus area, and it highlights a stroke of biological genius. You must pay attention to a structure called the mesosome.
The mesosome.
Since they lack internal membrane-bound organelles, prokaryotes utilize a specialized, differentiated form of the cell membrane itself. The mesosome is essentially an extensive infolding of the plasma membrane into the cytoplasm.
So instead of building a separate factory inside the city, they just heavily fold and convolute the outer border wall inward. The text mentions these extensions take the form of vesicles, tubules, and lamellae. It's a massive increase in surface area.
Precisely. By folding inward, they dramatically increase the surface area of the plasma membrane and proportionally increase the enzymatic content. Think of the mesosome as the ultimate multi-tool.
Okay.
It handles the heavy lifting tasks that eukaryotes delegate to distinct organelles. Mesosomes help in cell wall formation. They help in DNA replication and the intricate distribution of that duplicated DNA to daughter cells during division. They also manage respiration and secretion processes.
They are the prokaryotes' makeshift, all-in-one manufacturing and power plant.
That's the best way to think of it.
Surface area is the currency of biology. The more membrane real estate you have, the more enzymes you can embed to do work. The text also notes that in some specialized prokaryotes, like photosynthetic cyanobacteria, there are other membranous extensions into the cytoplasm called chromatophores, which hold the pigments.
Yes, it's the exact same principle of increasing functional surface area.
Now, what about mobility? If a bacterium needs to swim toward food or away from danger, how does it move?
If a bacterial cell is motile, it utilizes thin, filamentous extensions projecting from its cell wall called flagella. Bacteria show immense variation in the number and arrangement of these flagella.
What's the structure of the flagella?
Here's the structural breakdown you need to know. A bacterial flagellum is composed of three distinct parts: the filament, the hook, and the basal body. The filament is the longest portion, extending from the cell surface out into the environment, acting as the propeller.
Filament, hook, basal body, that is the motor for motility. But a lot of students get tripped up here because there are other structures sticking out of the bacteria that look like they should be for swimming but absolutely aren't.
Yes, this is a classic exam trap. Examiners will present a scenario and try to bait you into identifying pili or fimbriae as locomotor structures. They are not.
They are not.
While pili and fimbriae are indeed surface appendages, they play absolutely no role in motility.
So if they aren't propellers, what are they doing?
They are anchors and grappling hooks. The pili are elongated, tubular structures made of a special protein. The fimbriae are small, bristle-like fibers sprouting out of the cell surface.
I see.
In some bacteria, fimbriae are crucial for attaching the bacteria to rocks in fast-moving streams, or in the case of pathogenic bacteria, acting like Velcro to attach to host tissues. So flagella equals movement. Pili and fimbriae equal attachment. Do not confuse their functions.
Flagella for driving, pili and fimbriae for parking. That's perfectly clear. The last detail on the minimalist prokaryote is how they store resources. They don't have specialized fat cells or large vacuoles.
They rely on absolute simplicity, inclusion bodies. Reserve materials in prokaryotic cells are stored directly in the cytoplasm in the form of inclusion bodies.
And what's the key thing to remember about them?
The critical testable fact here is their naked state. They are not bounded by any membrane system whatsoever. They simply aggregate and lie free in the cytoplasm.
What kind of materials do they store?
Examples include phosphate granules, cyanophosphine granules, and glycogen granules. You also see specialized gas vacuoles in blue, green, purple, and green photosynthetic bacteria, which provide necessary buoyancy in water columns.
Naked storage granules just floating in the cytoplasmic fluid. It really completes the picture of the prokaryote, rugged, efficient, and minimalist. But now it is time to leave the frontier outposts. We are upgrading. we are moving into the sprawling, highly compartmentalized eukaryotic metropolis.
The shift from prokaryote to eukaryote is arguably the most significant architectural leap in evolutionary history. The eukaryotes encompass all the protists, plants, animals, and fungi.
The big leagues.
Exactly. The defining characteristic here is the extensive compartmentalization of the cytoplasm through a vast array of membrane-bound organelles. They possess a highly organized nucleus enclosed by a nuclear envelope. They utilize complex locomotory and cytoskeletal frameworks, and their genetic material is meticulously organized into complex chromosomes.
Before we dive into the specific organelles, it's worth noting that not all eukaryotic cities are zoned the same way. Plant and animal cells, for instance, have distinct differences based on their evolutionary lifestyles.
Yeah.
Plant cells are heavily fortified. They possess rigid cell walls, massive central vacuoles, and plastids like chloroplasts for solar power features completely absent in animal cells.
Conversely, animal cells maintain centrioles for division, which are absent in almost all higher plant cells.
Let's start at the city limits, the eukaryotic plasma membrane.
For a long time, the exact molecular architecture of the plasma membrane was a black box. The detailed structural view only emerged with the advent of the electron microscope in the 1950s.
But they knew something about it before that, right?
They did. Even before they could visually confirm it, brilliant biochemists used chemical studies on cell membranes, specifically utilizing human red blood cells, or erythrocytes, to mathematically deduce its structure.
Why the red blood cell? Oh, because mature mammalian red blood cells lack a nucleus and most internal organelles, making it much easier to isolate just the pure outer membrane for chemical analysis without contamination from internal membrane.
Exactly. It was a perfectly clean sample.
What did those studies reveal?
They revealed a highly specific ratio. Here is what you must remember, down to the exact percentage. The biochemical investigation proved that the cell membrane is primarily composed of lipids and proteins, with a small fraction of carbohydrates.
Right.
But specifically, in human beings, the membrane of the erythrocyte consists of exactly 52% protein and 40% lipids. Those numbers are a favorite for objective exams.
52% protein, 40% lipids. Got it. Now, how do those lipids actually assemble into a functional wall? The text focuses on the lipid bilayer. Let's build some intuition here because it's not a solid wall of brick. We need to talk about the thermodynamics of the lipid molecules themselves.
Yes, this is crucial. The major lipids involved are phospholipids, and they possess a very specific dual-natured orientation. We call them amphipathic.
Amphipathic.
They have polar, hydrophilic, or water-loving heads, and non-polar, hydrophobic, or water-fearing tails made of saturated hydrocarbons.
The inside of the cell is an aqueous, watery solution. The outside environment is also an aqueous, watery solution. So you have these lipid molecules where one half loves water and the other half is chemically repelled by it. How do they resolve that tension?
They self-assemble. The lipids spontaneously arrange themselves within the membrane so that their polar heads face outward, interacting directly with the aqueous environments on both the inside and outside of the cell.
And the tails.
Meanwhile, their hydrophobic tails tuck inward, facing each other, safely sandwiched in the middle. This bilayer arrangement ensures that the nonpolar tails are completely shielded from the aqueous environment. It is a brilliant, thermodynamically stable barrier.
So simple, yet so effective.
Alongside these phospholipids, you'll also find cholesterol embedded within the membrane to regulate its stability.
So we have an ocean of lipids forming the barrier. But what about that 52% protein content? Where do the proteins fit into this lipid ocean?
Depending on how deeply embedded they are, which determines the ease of biochemical extraction, membrane proteins are classified as either peripheral or integral. Okay.
Peripheral proteins lie loosely attached on the external surface of the membrane. Integral proteins, however, are partially or totally buried within the lipid bilayer itself.
If the lipid bilayer is the ocean, the peripheral proteins are like buoys or boats floating on the surface, while the integral proteins are massive icebergs plunging deep under the water, with some spanning the entire depth of the ocean to connect the inside of the cell to the outside.
I love that iceberg analogy.
This dynamic visual was finally formalized into a unifying theory, wasn't it?
Yes. The definitive, widely accepted model was proposed by Singer and Nicholson in 1972. It is famously known as the fluid mosaic model.
Singer and Nicholson, 1972. The fluid mosaic model. Let's decode that name. Why fluid and why mosaic?
It is a mosaic because the proteins are not uniformly distributed. They are scattered irregularly throughout the lipid bilayer, much like differently colored tiles in a mosaic artwork.
And fluid. And it is fluid because the lipid bilayer is not a rigid solid. It possesses a quasi-fluid nature. This quasi-fluidity enables the lateral movement of proteins within the overall bilayer.
They aren't frozen in place.
No, they can drift and navigate through the lipid sea. The ability of these proteins and lipids to move laterally within the membrane is how we measure the membrane's fluidity.
So it's a living, shifting, dynamic border, and that fluidity is strictly necessary, isn't it? If the membrane were a solid sheet of rigid plastic, the cell couldn't function.
Exactly. The fluid nature of the membrane is an absolute physiological requirement. It is critical for functions like cell growth, the formation of intercellular junctions, secretion, endocytosis, where the membrane physically engulfs outside material, and, crucially, cell division.
Right, a rigid boundary could never pinch inwards to divide one cell into two.
It would just shatter.
That brings us to the primary job of this fluid border: transport and customs. The membrane is selectively permeable. It tightly regulates what enters the metropolis and what exits. Let's look at the physics of how things cross. The most basic method is passive transport.
Passive transport is the movement of molecules across the membrane without any expenditure of metabolic energy. The cell doesn't have to pay a toll.
Free entry.
Yes. For instance, neutral nonpolar solutes can slip right through the lipid bilayer via simple diffusion. They move along the concentration gradient, spontaneously diffusing from an area of higher concentration to an area of lower concentration, driven purely by entropy.
The universe naturally wants to balance things out. If there's a lot of oxygen outside the cell and very little inside, the oxygen naturally diffuses in.
Exactly.
And when the solvent itself, water, moves across the membrane along its concentration gradient, we give that specific diffusion a name: osmosis.
Right.
Both simple diffusion and osmosis are free. But what happens when a molecule isn't neutral? What if it's highly polar?
This is where the thermodynamics of the hydrophobic tails pose a problem. Highly polar molecules, or ions, cannot spontaneously cross the nonpolar lipid interior. The hydrophobic tails will forcefully repel them.
So they need help.
Therefore, these polar molecules require specialized infrastructure. They need a carrier protein, an integral protein acting as a dedicated channel or gate to facilitate their transport across the membrane. This is still passive as it flows down the gradient, but it requires structural facilitation.
And finally, what if the city needs to import something that is already highly concentrated inside, or export waste that is highly concentrated outside? What if it has to push against the natural flow of the universe?
That requires work. A few ions or molecules must be transported across the membrane against their concentration gradient from a region of lower concentration to a region of higher concentration.
Uphill, basically.
Yes. Such transport is an energy-dependent process. The cell must spend its metabolic currency, ATP, to physically pump the molecules across. This is called active transport.
Active transport.
The textbook example, which is fundamental to neurobiology, is the sodium-potassium pump. Active transport requires ATP. Passive transport does not.
So active transport is the cell taxing its energy reserves to maintain an artificial chemical imbalance, which is the very definition of being alive.
Brilliantly stated.
Now, before we venture deeper into the cytoplasm, we need to briefly acknowledge the cell wall, which sits external to the plasma membrane in fungi and plants. It's a non-living, rigid structure.
Very important structure.
It provides structural shape, defends against mechanical stress and pathogenic infection, and acts as a coarse filter against massive, undesirable macromolecules.
The chemical composition of the cell wall is highly specific depending on the organism. In algae, the cell wall is composed of cellulose, galactans, mannans, and minerals like calcium carbonate.
And in higher plants?
In higher plants, it consists of a complex matrix of cellulose, hemicellulose, pectins, and proteins. You should also understand its developmental trajectory.
How so?
In a young plant cell, the primary wall is thin and capable of growth. As the cell matures, this growth capacity diminishes, and a much thicker secondary wall is laid down on the inner side, directly adjacent to the plasma membrane.
If every plant cell is encased in a rigid box, how do they form a cohesive tissue? How do they stick together?
Neighboring plant cells are cemented together by a layer called the middle lamella, which is primarily composed of calcium pectate. It functions as the mortar between the bricks.
The mortar, okay.
Furthermore, to prevent these cells from being completely isolated, the cell wall and middle lamellae are traversed by microscopic channels called plasmodesmata.
Plasmodesmata.
These channels directly bridge the cytoplasm of neighboring cells, allowing for the rapid exchange of materials and chemical signals.
The plasmodesmata are like secret underground tunnels connecting adjacent walled cities.
That's a great image.
Okay, we have cleared the borders. We are standing inside the eukaryotic metropolis. The first thing we realize is that the manufacturing sector of this city isn't just a random collection of factories. Several massive organelles are physically and functionally linked together into a single, coordinated production line.
The text refers to this as the endomembrane system.
Yes. While the eukaryotic cell contains many distinct membranous organelles, several of them are considered together as the endomembrane system because their physiological functions are intimately coordinated. They operate as a continuous logistical pathway.
Like an automotive assembly line.
Exactly. You have different massive machines, but they are all working sequentially on the same product, from raw material to final shipping. Which organelles officially make up this coordinated endomembrane system?
The endomembrane system strictly includes 4 components: the endoplasmic reticulum, the ER, the Golgi complex, lysosomes, and vacuoles.
Now, here is where examiners love to see if you're paying attention. There are massive organelles that are explicitly not part of this system.
Here is what you must remember. Mitochondria, chloroplasts, and peroxisomes are not considered part of the endomembrane system.
It's a classic trick.
A very common multiple-choice question will present a list of organelles and ask you to identify the odd one out regarding the endomembrane system.
And why are they excluded?
The reason these 3 are excluded is that their functions, energy generation, and specific oxidative reactions are completely independent and not coordinated with the interconnected manufacturing and packaging pathway of the ER, Golgi, lysosomes, and vacuoles.
Excellent. Let's walk the assembly line, starting at the beginning. The endoplasmic reticulum, or ER. Under an electron microscope, it looks like an incredibly vast network or reticulum of tiny tubular structures scattered throughout the cytoplasm.
Because it's such an expansive network of folded membranes, the ER physically divides the intracellular space into 2 distinct environments.
Right.
It creates a stark compartmentalization. You have the luminal compartment, which refers to the protected space inside the folds of the ER. And then you have the extraluminal compartment, which is the surrounding cytoplasm.
And there are 2 types of ER, based on what they look like, right?
Yes. Based on its surface topology under a microscope, the ER is divided into 2 distinct functional zones. Often, you will see the ER heavily studded with ribosomes attached to its outer surface. This gives it a granular appearance, which we call the rough endoplasmic reticulum, or RER.
Rough because it looks like a microscopic cobblestone street. And since we know ribosomes are the machines that synthesize proteins, the rough ER must be the heavy industry zone for protein manufacturing.
Precisely. RER is extensive and frequently observed in cells that are aggressively involved in protein synthesis and subsequent secretion, like pancreatic cells producing digestive enzymes.
Excellent.
Furthermore, the architecture of the RER is functionally continuous with the outer membrane of the nuclear envelope. This allows the messenger RNA coming out of the nucleus to immediately dock with the ribosomes on the RER to begin translation.
It's extremely efficient.
Can vary. Conversely, regions of the ER where ribosomes are completely absent appear smooth. This is the smooth endoplasmic reticulum, or SER.
If the smooth ER lacks the protein-building ribosomes, what is its designated role on the factory line?
The smooth endoplasmic reticulum shifts its focus to lipid chemistry. It is the major intracellular site for the synthesis of lipids. In animal cells specifically, the SER is responsible for synthesizing crucial lipid-like steroidal hormones.
So the rough ER manufactures the complex proteins and the smooth ER manufactures the lipids and steroids.
Correct.
Once these raw products are synthesized, they can't just be dumped into the cytoplasm. They need to be chemically modified, sorted, and shipped to their final destinations. That brings us to the logistics hub, the Golgi Apparatus.
First observed by the Italian scientist Camillo Golgi in 1898. He identified densely stained reticular structures localized near the nucleus.
What does it look like?
The Golgi apparatus consists of a series of highly organized, flat, disc-shaped sacs, or cisternae, ranging from 0.5 to 1.0 micrometers in diameter. These cisternae are stacked densely parallel to each other, resembling a stack of hollow pancakes.
The physical orientation of the Golgi is crucial to its function, though. It's not perfectly symmetrical. The text emphasizes that it's highly polarized, possessing 2 distinct faces.
Yes, the polarity of the Golgi is central to understanding its mechanism. You must be familiar with the nomenclature.
Let's hear it.
There is the convex forming face, which is structurally designated as the cis face. Then, pointing away, there is the concave maturing face, designated as the trans face.
Cis and trans.
The cis and trans faces are entirely different structurally, yet they are functionally interconnected by the flow of the cisternae between them.
Let's trace the flow of product. A newly minted protein leaves the endoplasmic reticulum. What happens next?
The Golgi apparatus principally performs the function of packaging these raw materials, preparing them to be delivered either to specific intracellular targets or to be secreted entirely outside the cell.
Okay, packaging.
The materials from the ER are enclosed in transport vesicles. These vesicles physically fuse with the cis-forming face of the Golgi. From there, the material is passed deep into the cisternae, moving systematically towards the maturing trans face.
Which makes sense why the Golgi is right next to the ER.
Exactly. This requisite directional flow elegantly explains why the Golgi apparatus is always found in close physical association with the endoplasmic reticulum.
It catches the cargo with the cystoc, processes it as it moves to the warehouse, and ships it out the transloading bays. What exactly constitutes processing in this context?
A significant number of proteins synthesized by ribosomes on the ER undergo vital chemical modifications within the cisternae of the Golgi before they are cleared for release from the trans phase.
Such as?
Most notably, the Golgi apparatus is the primary site for glycosylation, the complex addition of carbohydrate groups to proteins and lipids, resulting in the formation of functional glycoproteins and glycolipids. These molecules are essential for cell-to-cell recognition and signaling.
So ER synthesizes, Golgi modifies, and dispatches. Now, what happens to the byproducts? What happens when the cell needs to break down obsolete machinery or digest large food particles? We move to the third component of the endomembrane system, lysosomes.
Lysosomes are the dedicated waste management and recycling facilities. They are membrane-bound vesicular structures that are physically formed by the packaging process in the Golgi apparatus. They literally bud off from the trans face.
And what's inside them?
When isolated and analyzed, these lysosomal vesicles are found to be densely packed with an incredibly aggressive arsenal of hydrolytic enzymes collectively called hydrolases. This includes lipases, proteases, and carbohydrases.
Lipases cleave lipids, proteases dismantle proteins, carbohydrases break apart carbohydrates. They are effectively microscopic bags of acid and digestive enzymes capable of breaking down nearly every biological macromolecule.
That's exactly what they are.
Which raises an immediate engineering question. If they're so universally destructive, why don't they accidentally digest the cell itself?
This reveals a breathtaking level of biochemical fail-safes. The destructive capacity of these enzymes is entirely dependent on pH. These specific hydrolytic enzymes are optimally active only at a highly acidic pH.
Oh, I see.
The lysosome actively pumps protons into its lumen to maintain an internal acidic environment far below the neutral pH of the surrounding cytoplasm.
So if it breaks?
If a lysosome were to be structurally compromised and accidentally leak its enzymes into the cytoplasm, the sudden shift to a neutral pH would instantly render the enzymes inactive, sparing the cell from catastrophic autodigestion.
That is just phenomenal biological engineering. The weapon is disabled if it ever leaves the designated firing range. Finally, rounding out the endomembrane system, we have the vacuoles. We touched on these when discussing plant cells, but their role is expansive.
The vacuole is a distinct membrane-bound space found within the cytoplasm. It acts as a massive reservoir containing water, dissolved sap, excretory products, and various other materials that are not currently required by the active cytoplasm.
What's a membrane called?
The vacuole is bounded by a single specialized membrane. You must commit the name of this membrane to memory. It is called the tonoplast.
The tonoplast. And in the context of plant cells, these are not just minor storage closets. The text highlights a mind-blowing statistic regarding their sheer volume.
In mature plant cells, the central vacuole can expand to occupy up to 90% of the total volume of the cell.
90%.
It pushes all the other organelles to a thin peripheral layer against the cell wall. And the tonoplast is not a passive barrier. In plants, the tonoplast actively expends energy to transport a vast array of ions and other materials against their concentration gradients into the vacuole.
Active transport again.
Yes. Consequently, the concentration of these materials is maintained significantly higher inside the vacuole than in the cytoplasm, creating high turgor pressure that keeps the plant rigid.
Beyond just plant structure, vacuoles adapt to their organism. In an amoeba, the contractile vacuole is critical for osmoregulation and excretion, acting like a bilge pump to constantly expel excess water so the single-celled organism doesn't swell and burst.
Right. And in many protists, specialized food vacuoles are formed dynamically by engulfing food particles for intracellular digestion.
That completes our tour of the endomembrane system, the beautifully orchestrated linear pathway of the ER, Golgi, lysosomes, and vacuoles. But now, we must turn our attention to the entities that we explicitly excluded from that system, the heavy industry powerhouses.
We are entering the domain of the energy titans, the mitochondria and the plastids.
And I have to be honest, reading the deep literature on these specific organelles triggered the most profound whoa moment I've had in a long time. It introduces the staggering concept of cells living inside of other cells. Let's start with the universally famous powerhouse, the mitochondrion.
Mitochondria are fascinating. Unless they are specifically treated with targeted stains, they are nearly invisible under a standard light microscope. Furthermore, their sheer number per cell is not fixed.
It changes.
It is highly variable and directly correlates with the physiological activity and energy demands of that specific cell type. A highly active muscle cell or liver cell will possess thousands of mitochondria, whereas a dormant cell will have far fewer. Morphologically, they are typically sausage-shaped or cylindrical.
Let's dissect their architecture. Unlike the single membrane vacuoles or lysosomes, the mitochondrion boasts a double membrane structure. It has a smooth outer membrane and a highly complex inner membrane.
These two membranes divide the organelle's lumen into two distinct aqueous environments, the outer compartment and the inner compartment.
And what's inside the inner compartment?
The inner compartment is filled with a dense, biochemically rich, homogeneous substance called the matrix.
The matrix.
The outer membrane serves as the continuous limiting boundary, controlling basic access. But the inner membrane is where the sophisticated biochemical machinery resides. To maximize its functional capacity, the inner membrane forms a multitude of intricate infoldings directed inwards toward the matrix. These specific infoldings are called cristae.
We've seen this exact physical strategy before. Just like the mesosome folded inward in the prokaryotic bacteria to maximize real estate, the cristae fold inward to dramatically increase the surface area of the inner mitochondrial membrane.
The principle of surface area is universal. The two distinct membranes each possess their own highly specialized repertoire of enzymes associated with specific stages of mitochondrial function.
That's their main job.
Mitochondria are the exclusive sites of aerobic respiration in the eukaryotic cell. They harness oxygen to extract energy from macromolecules, ultimately producing cellular energy in the form of ATP adenosine triphosphate. This is why they earn the ubiquitous title of the powerhouses of the cell.
Okay, here is where my mind was absolutely blown. If you look closely at the dense matrix inside that inner membrane, it isn't just a soup of respiratory enzymes.
No, it is not.
The text explicitly points out that the matrix also possesses a single double-stranded circular DNA molecule. It has a few RNA molecules, it has its own ribosomes, it has all the independent components required for the synthesis of its own proteins. And, perhaps most shockingly, mitochondria divide and multiply on their own timetable via independent fission.
I want you to connect the dots here. Think about what you just listed. An entity that possesses a single circular DNA molecule, manufactures its own proteins, and replicates through binary fission. Does that biological profile sound familiar to you?
It sounds identical to the prokaryotic bacteria we discussed in Section 2.
Yeah.
It's an unavoidable conclusion. It looks exactly like an ancient eukaryotic cell swallowed a primitive oxygen-breathing bacteria a billion years ago, and instead of digesting it in a lysosome, struck an evolutionary deal. The eukaryote provided a safe, nutrient-rich environment, and the bacteria provided massive amounts of ATP. They became inseparable. It's an alien organism living permanently inside our cells.
You are describing the fundamental premise of the endosymbiotic theory. While modern textbooks might introduce it gradually, the structural evidence they present is overwhelming. And for our high-achieving listeners, here is a highly testable, neat focus point that seals the deal on this theory.
The ribosomes.
Yes. We established earlier that the vast cytoplasm of a eukaryotic cell utilizes ADS ribosomes for protein synthesis. But if you extract the ribosomes found exclusively inside the mitochondrial matrix, what size are they?
The text states definitively, the ribosomes inside the mitochondria are 70S. They match the exact dimensions and sedimentation coefficient of the ribosomes found in independent, free-living prokaryotic bacteria.
Exactly. You must cement that fact in your memory. Mitochondria possess 70S ribosomes, in stark contrast to the 80S ribosomes found in the surrounding eukaryotic cytoplasm. This distinction is one of the most frequently tested concepts in cell biology.
Unbelievable. Now let's look at the other major energy titans, which share a similarly fascinating origin story. The plastids.
Plastids are massive organelles found exclusively in all plant cells and in euglenoids. They're large enough to be easily observed under a light microscope.
What makes them special?
Their primary defining feature is that they bear specific chemical pigments, which impart the vibrant colors we associate with plant life. Based on the specific type of pigments they contain and consequently their biological function, plastids are broadly classified into three main categories: chloroplasts, chromoplasts, and leucoplasts.
Let's break down the functional distinctions. First, the most critical for global ecology.
Yeah.
The chloroplasts.
Chloroplasts contain massive amounts of chlorophyll and varying amounts of carotenoid pigments. These are the specialized molecules responsible for trapping and harnessing the raw light energy essential for driving the chemical reactions of photosynthesis.
Next, we have the chromoplasts. The prefix chromo relates directly to color.
In chromoplasts, fat-soluble carotenoid pigments, specifically keratin and xanthophylls, are synthesized and stored. These pigments are responsible for imparting the brilliant yellow, orange, or red colors to specific parts of the plant.
Like leaves in the fall.
Think of the vibrant red of a ripe tomato, the deep orange of a carrot, or the spectacular, fiery display of autumn leaves. That is all the work of chromoplasts.
Finally, we have the leucoplasts. Leuco translates to white, or more accurately in this context, colorless.
Leucoplasts are the large, colorless plastids found in varied shapes and sizes. Because they lack photosynthetic pigments, their primary physiological role is the long-term storage of vital nutrients.
And there are three types of these too.
You are required to know the three specific subtypes of leucoplasts based on what they store. Amyloplasts are responsible for synthesizing and storing large carbohydrates, specifically starch, as seen abundantly in potatoes. Elaioplasts specialize in the storage of complex oils and fats, and aleuroplasts are dedicated to the storage of proteins.
Okay, let's execute a deep dive into the architecture of the most famous plastid, the chloroplast. If we look at a green leaf, where are these concentrated?
The vast majority of the chloroplasts in green plants are densely concentrated within the mesophyll cells of the leaves. Morphologically, they can be lens-shaped, oval, discoid, or even ribbon-like.
And double membrane again.
Much like the mitochondria, chloroplasts are bounded by a double membrane envelope. Of these two membranes, the inner chloroplast membrane is relatively less permeable, acting as a tighter regulatory gate. The massive, fluid-filled inner space limited by this inner membrane is called the stroma.
So the stroma in a chloroplast is the direct equivalent of the matrix in a mitochondrion. It's the primary workspace. And just like the inner membrane of the mitochondria folded into cristae to maximize its operational surface area, the chloroplast deploys its own internal architectural system inside the stroma to maximize its capacity for catching sunlight.
The internal architecture of the chloroplast is a marvel of spatial efficiency. Suspended within the stroma is a highly organized network of flattened membranous sacs called thylakoids.
Thylakoids.
The vital chlorophyll pigment, the actual solar receptors, are embedded directly within the membranes of these thylakoids. To maximize light absorption efficiency in a limited volume, these thylakoids do not float randomly. They're stacked precisely on top of one another, much like piles of coins.
And what's a stack called?
An entire stack is referred to as a granum or intergranal thylakoids.
So a single flattened solar panel is a thylakoid. A tightly packed vertical array of these panels is a granum. But are these individual towers isolated from one another?
No, the system is fully interconnected. In addition to the stacks, there are flat membranous tubules called stroma lamellae.
Stroma lamellae.
These lamellae act as bridges, physically connecting the thylakoids of adjacent grana across the stroma. It forms a continuous, highly structured internal grid. Furthermore, the membrane of each thylakoid encloses a hollow inner space called a lumen, which is crucial for building the proton gradients required to generate energy.
And what is occurring in the surrounding stroma fluid? While the thylakoids are catching the light, what is the fluid doing?
The stroma is far from empty space. It contains the complex array of enzymes required for the synthesis of carbohydrates and proteins. This is where the cell utilizes the energy trapped by the thylakoids to literally build sugar molecules out of thin air during the dark reactions of photosynthesis.
But bringing it back to our earlier revelation regarding cells within cells, what else resides inside the stroma?
This is the kicker. Alongside those sugar-building enzymes, the stroma contains small, double-stranded circular DNA molecules. It contains its own ribosomes. And just like the mitochondria, the text explicitly mandates we remember. The The ribosomes of the chloroplasts are smaller, measuring at 70s, completely distinct from the 80s cytoplasmic ribosomes of the host eukaryotic cell.
Another instance of 70S ribosomes, another piece of undeniable structural evidence pointing to an independent prokaryotic origin.
Absolutely. So, to tightly summarize the energy titans for our learners, both mitochondria and chloroplasts are fortified by double membranes. Both are dedicated to profound energy conversion, producing ATP and building sugars, respectively. Both possess their own independent circular DNA. And crucially, both utilize 70S ribosomes, diverging from the standard eukaryotic 80S model.
It's an incredible biological reality. We've now mapped the borders, the industrial manufacturing lines, the recycling plants, and the independent power grids. Now we must examine the physical scaffolding that prevents the city from collapsing, the motors that drive its movement, and finally, the central vault that commands it all. Let's begin by returning to those ribosomes we've been tracking. They are the non-membrane-bound organelles found ubiquitously across all forms of life.
Ribosomes are the universal translators of life. Let's review their history and the specific mathematics that governs their classification. They are dense, granular structures that were first clearly observed under the electron microscope by George Pollard in 1953.
Made of what?
Biochemically, they are composed of ribonucleic acids, specifically rRNA and proteins. And importantly, they are completely naked. They are not surrounded by any lipid membrane.
The literature gives us some specific math regarding their structural subunits. We established that eukaryotic ribosomes are 80S, while prokaryotic, mitochondrial, and chloroplast ribosomes are 70S. Before we look at the parts, what exactly does that S represent?
The S stands for Svedberg's unit. It is not a direct measurement of weight. Rather, it represents the sedimentation coefficient.
What does it mean?
It is an empirical measure of how rapidly a given particle settles down to the bottom of a test tube when subjected to extreme gravitational forces in an ultracentrifuge. Because sedimentation speed is influenced by both mass and the aerodynamic or hydrodynamic shape of the particle, the Svedberg unit is an indirect measure encompassing both density and size.
Down it.
Now, functionally, every complete ribosome is constructed from two distinct interlocking subunits, a larger domed unit and a smaller cap unit.
Here's where the math feels completely counterintuitive to a student, so let's clarify the physics of it.
Yes, if you look at the eukaryotic 80S ribosome, its two individual subunits are measured at 60S for the large subunit and 40S for the small subunit.
But 60 plus 40 equals 100.
Exactly. Obviously, 60 plus 40 equals 100, not 80. This is because sedimentation coefficients are not strictly additive. When the 60S and 40S subunits lock together, their combined, more streamlined shape creates more drag than their individual masses would suggest, so the complete assembly sediments at an 80S rate.
Ah, I see. And the prokaryotic ones?
Similarly, the prokaryotic 70S ribosome is composed of a 50S large subunit and a 30S small subunit. Again, 50 plus 30 equals 80, but the combined complex sediments at 70S, you must memorize these specific subunit pairings. 80S is 60S plus 40S, 70S is 50S plus 30S.
The physics of drag altering the math. It makes perfect sense once you realize S isn't just mass. Next, let's address the structural integrity of the cell. If you were to extract all the water, a eukaryotic cell does not just collapse into a shapeless puddle like a deflated balloon. It maintains a highly structured internal 3D architecture.
This internal scaffolding is called the cytoskeleton. It is an incredibly elaborate dynamic network of filamentous proteinaceous structures that pervade the entire cytoplasm.
What's it made of?
It primarily consists of three distinct types of filaments: microtubules, microfilaments, and intermediate filaments. The cytoskeleton is not merely structural, it is heavily involved in a myriad of critical physiological functions.
Like what?
It provides immense mechanical support to resist compression and tension. It coordinates intracellular motility, acting as the highway system for motor proteins carrying vesicles, and it meticulously maintains the overall shape of the cell.
Microtubules, formed from the protein pobulin, are a particularly vital component of that cytoskeleton, and these massive microtubule beams are not just used for internal scaffolding. The cell extrudes them outwards to build the complex motors of the city, the cilia and flagella. We discussed the minimalist prokaryotic flagella earlier. Are the eukaryotic versions built the same way?
They are completely structurally distinct. The eukaryotic cilia and flagella are vastly more complex. They are hair-like outgrowths of the cell membrane.
Cilia act like oars, right?
Cilia are relatively small structures which operate mechanically like oars, causing the coordinated movement of either the cell itself or the fluid surrounding the cell, such as the cilia in our respiratory tract clearing mucus.
And flagella?
Flagella are comparatively much longer, whip-like structures and are primarily responsible for propelling the entire cell forward, as seen in a spermatozoon.
The text provides a transverse electron microscopic breakdown of a eukaryotic cilium, or flagellum. It emphasizes that they are entirely covered by the continuous plasma membrane. So they aren't naked appendages glued to the outside. They are complex structural extensions forcefully pushed out from inside the membrane. The core structural engine of this extension is called the axoneme.
Now, I need everyone listening to lean in. Prepare for a highly specific structural axoneme.
Give us the layout.
The axoneme possesses a complex array of microtubules running parallel to its long axis. Specifically, it features 9 distinct doublets of radially arranged peripheral microtubules forming an outer ring and a separate pair of centrally located single microtubules.
Let's visualize that carefully. 9 peripheral doublets. So that's 18 individual microtubules arranged in paired sets around the outer perimeter, and then 2 distinct individual microtubules running right down the dead center.
Exactly. This highly conserved structural geometry is universally referred to as the 9+2 array. It is a masterpiece of mechanical engineering.
How does it all hold together?
The 2 central tubules are connected to each other by structural bridges and are entirely enclosed by a protective central sheath. The central sheath is then physically connected to one of the tubules of each of the 9 peripheral doublets by a radial spoke.
So 9 spokes.
Consequently, there are exactly 9 radial spokes holding the central core to the outer ring. Furthermore, the peripheral doublets are not just floating, they're actively interconnected by proteinaceous linkers. This entire sliding microtubule complex is what generates the whipping motion of the flagellum.
So cilia and flagella utilize this complex 9+2 array of microtubules. Now, these massive structures don't just magically appear. Where do they grow from? The text indicates that both the cilium and flagellum emerge from a foundational anchor structure called the basal body, which is structurally identical to a centriole. Which forces us to ask, what exactly is a centriole?
Let's transition to the centrosome and the centrioles. A centrosome is an organelle typically found in animal cells, heavily involved in cell division. It usually contains 2 distinct cylindrical structures known as centrioles.
2 centrioles in a centrosome.
These centrioles are surrounded by a cloud of amorphous pericentriolar materials. Interestingly, the 2 centrioles within a centrosome always position themselves to lie exactly perpendicular to each other, forming an L shape.
And what does their internal microtubule architecture look like? Are they just buried versions of the 9+2 array?
They absolutely are not, and this structural distinction is precisely what examiners will test you on. Each individual centriole possesses an organization that resembles a cartwheel.
A cartwheel?
They are constructed from 9 evenly spaced peripheral fibrils of tubulin protein. However, each of these peripheral fibrils is a triplet, not a doublet. There are 9 sets of 3 microtubules around the outside.
And what's in the middle?
Furthermore, the center is completely empty of microtubules. The central part of the proximal region of the centriole is solid protein, called the hub. This central hub is connected to the peripheral triplets by thick radial spokes made of protein.
So to contrast them directly, centrioles feature a 9+0 array of peripheral triplets with a protein hub. Cilia and flagella feature a 9+2 array of peripheral doublets with 2 central microtubules.
Precisely. You must keep those numbers distinct. The centrioles have 2 massive responsibilities in the animal cell. First, they migrate to the membrane to form the basal body, acting as the seed from which cilia or flagella grow.
And
Second, and crucially, during cell division, they dictate the formation of the spindle fibers that give rise to the massive spindle apparatus, which physically pulls the chromosomes apart to opposite poles of the dividing cell.
And speaking of pulling apart chromosomes, we have finally arrived at the absolute center of the metropolis, the master archive, the central command, the nucleus. It was first explicitly described as a distinct cell organelle by Robert Brown way back in 1831. Much later, when scientists began using basic dyes, the highly stainable material packed inside the nucleus was named chromatin by Walther Fleming.
Let's examine the interphase nucleus, which represents the nucleus of a cell when it is in its normal metabolic state, not actively undergoing division. It is highly structured.
What is inside?
It features a highly extended, incredibly elaborate network of nucleoprotein fibers called chromatin, an internal fluid matrix called the nucleoplasm, and one or more prominent spherical bodies known as nucleoli.
The perimeter of the nucleus is heavily fortified to protect the genetic blueprints. The nuclear envelope actually consists of two parallel membranes. There is a distinct measurable space between them, ranging from 10–50 nanometers wide, known as the paranuclear space.
This double membrane system forms a rigorous barrier between the sensitive genetic materials present inside the nucleus and the highly active, potentially hazardous metabolic environment of the cytoplasm.
The outer membrane of this nuclear envelope usually remains physically continuous with the rough endoplasmic reticulum we discussed earlier, and correspondingly, it often bears ribosomes on its cytoplasmic surface.
Yeah.
However, a master vault is useless if the blueprints can never be sent out to the factory floor. The barrier is not completely sealed. At a multitude of specific locations, the nuclear envelope is interrupted by minute, highly complex structures called nuclear pores. These pores are formed by the intricate fusion of the inner and outer membranes.
What exactly is passing through these heavily guarded nuclear pores?
I'm going to ask you the same thing.
These nuclear pores act as the highly regulated customs checkpoints. They are the specific bidirectional passages through which the movement of massive RNA molecules and specific regulatory proteins takes place between the nucleus and the cytoplasm.
So the DNA never leaves.
The master DNA blueprint never leaves the vault, but the messenger RNA copies are shipped out through these pores to the ribosomes, while essential proteins synthesized in the cytoplasm are imported back in.
And what about the dense spherical bodies inside? The nucleoli. The singular is nucleolus. The text makes a specific point that the nucleolus is not bounded by any internal membrane. Its dense granular content is completely continuous with the surrounding nucleoplasm. What is happening in that dense region?
It is a massive, highly active factory operating directly inside the vault. The nucleolus is the dedicated site for the active synthesis of ribosomal RNA, or rRNA. Recall the thousands of ribosomes required by the rough ER.
Yeah.
The core components of those machines are manufactured right here. Consequently, larger and far more numerous nucleoli are predictably present in specific cells that are actively engaged in massive amounts of protein synthesis, as they require a constant massive supply of new ribosomes to sustain that production.
Now let's address the core data storage itself, the DNA. You mentioned the term chromatin. During normal interphase, the DNA exists as this loose, indistinct, tangled network of nucleoprotein fibers. It looks like a massive bowl of microscopic spaghetti.
Right.
But when the cell receives the signal that it is time to divide, it cannot safely separate a tangled mess. It has to pack that data up into tight, transportable units.
Yes, the transition from chromatin to chromosomes is a miracle of biological packaging. During the different stages of cell division, cells no longer show a diffuse nucleus. Instead, they display highly structured, aggressively condensed chromosomes.
The chromatin material itself is a complex matrix. It contains the massive double helix of DNA, but also specific basic proteins called histones that act as spools for the DNA to wind around. It also contains some non-histone proteins and structural RNA.
To give you a genuine sense of the staggering packaging required, a single microscopic human cell contains approximately 2 full meters of DNA thread.
2 meters in one cell.
That 2 meters of material must be meticulously distributed and tightly folded into 46 distinct chromosomes, representing our 23 pairs. 2 meters of data compressed into a space measured in micrometers.
It is an incomprehensible feat of geometry and physics. To help you build an accurate mental map of these transportable data drives, the text outlines the basic anatomy of a chromosome. Every distinctly visible chromosome essentially features a primary constriction, a pinched waistline area.
This vital anchor point is called the centromere, and flanking the sides of the centromere are highly specialized disc-shaped structures.
What's this called?
Those dense, disc-shaped protein structures on the sides are called kinetochores. The kinetochores serve as the physical attachment points for the spindle fibers generated by the centrioles we discussed during cell division.
And the centromere.
The centromere itself physically holds the 2 identical sister chromatids of a duplicated chromosome tightly together until it is time to rip them apart. Now, here is the final major structural classification scheme you must absolutely master for Neet exams.
Based strictly on the physical position of that centromere along the length of the chromosome, Chromosomes are morphologically classified into 4 distinct types. Visualizing this is crucial. Imagine a chromosome as 2 arms extending out from a central knot. By sliding that knot up or down, you alter the relative lengths of the arms.
Let's methodically run through these 4 types. Visualize the geometry. The first type is the metacentric chromosome.
Where's the knot?
The metacentric chromosome has its centromere located exactly in the dead middle. This perfectly central positioning divides the chromosome into 2 completely equal arms. The prefix meta implies middle. During cell division, as it is pulled by the center, it bends into a perfect V shape.
Second is the submetacentric chromosome.
In the submetacentric chromosome, the centromere is shifted slightly away from the exact middle of the chromosome. This asymmetrical placement results in one visibly shorter arm and one correspondingly longer arm. When pulled, it forms an L shape.
Third is the acrocentric chromosome.
The acrocentric chromosome pushes this asymmetry to the extreme. The centromere is situated very close to its terminal end. This configuration forms one extremely short, stubby arm and one exceptionally long arm. It resembles a J shape during division.
And finally, the 4th type is the telocentric chromosome.
The telocentric chromosome has a truly terminal centromere. The knot is situated right at the absolute tip, meaning it essentially possesses only one set of arms extending downward. The prefix telo relates to the end or tip.
So metacentric is middle, submetacentric is slightly off-center, acrocentric is near the end, and telocentric is exactly at the end.
Perfect.
Metacentric, submetacentric, acrocentric, telocentric, the V, L, J, and I shapes. Now, the text briefly mentions one final tiny structural quirk present on some specific chromosomes. Sometimes, a select few chromosomes possess completely non-staining secondary constrictions at a constant, highly specific location along their length. What does that visually result in?
Because that secondary constriction pinches the chromatin so tightly, it gives the visual appearance of a small, distinct, bulbous fragment attempting to break off the end of the main chromosome. This small terminal fragment is scientifically termed a satellite. Chromosomes bearing these are often referred to as SAT chromosomes, and they play specific roles in nucleolus formation.
And with that final detail regarding the satellite, we have comprehensively mapped the entire span of the eukaryotic metropolis. From the sticky defensive Glycocalyx of a humble bacteria through the active customs gates of the plasma membrane, down the highly coordinated assembly lines of the endomembrane system, past the independent prokaryotic powerhouses trapped in the matrix, along the intricate 9+2 scaffolding, and finally deep into the master genetic vault of the nucleus.
It's an epic journey. Oh, and for the sake of absolute completeness, the text briefly notes that there are numerous other membrane-bound minute vesicles called microbodies that contain various specialized oxidative enzymes, which are abundantly present in both plant and animal cells. They represent just another granular level of the city's compartmentalization strategy.
We have covered an absolutely incredible foundational amount of ground today. It is time to lock it all down. I'm calling for the sticky summary. If you only walk away with a handful of absolute certainties, what are the top 5 critical facts to dominate this topic on an exam?
I will gladly provide the anchor points. Here's what you must remember.
Fact 1.
Regarding cell theory, Schleiden the botanist and Schwann the zoologist laid the foundation, but it was Virchow who fundamentally completed it with the phrase omnis cellula cellula, proving that all new cells exclusively arise from pre-existing cells.
Fact 2.
When dealing with prokaryotic biology, remember their specialized tools. Plasmids confer external powers like antibiotic resistance. Mesosomes are inward membrane folds acting as makeshift factories for respiration and replication. And inclusion bodies provide entirely naked storage within the cytoplasm.
Fact 3.
The endomembrane system is a heavily coordinated logistical pathway that strictly includes the ER, Golgi, Lysosomes, and Vacuoles. You must remember that mitochondria, chloroplasts, and peroxisomes are definitively excluded from this system.
Fact 4.
Ribosome mathematics is highly testable. Eukaryotic cytoplasm utilizes 80S ribosomes constructed from 60S and 40S subunits. In stark contrast, prokaryotes, as well as the interior matrix of mitochondria and the stroma of chloroplasts, utilize 70S ribosomes constructed from 50S and 30S subunits.
And fact 5.
Microtubule geometry is a favorite trap. Cilia and flagella utilize a 9+2 array of peripheral doublets for motility, whereas centrioles and basal bodies utilize a 9+0 array of peripheral triplets arranged around a central protein hub.
That summary is pure, concentrated gold. To close out our deep dive today, I want to leave you, our listener, with a final, slightly provocative thought, directly inspired by the boundaries we established at the very beginning of the session. We started by declaring that anything less than a complete structure of a cell does not ensure independent living. We also established that the nucleus, with its 2 meters of DNA, is the absolute master brain controlling the cell's activities and future. However, the text explicitly points out an anomaly.
A fascinating anomaly.
Some highly mature, specialized cells, specifically the functional erythrocytes or red blood cells of many mammals, as well as the vital sieve tube cells responsible for nutrient transport in vascular plants, completely lack a nucleus. They eject it to maximize space. So, if we adhere strictly to our physicochemical definitions, can an entity that has completely jettisoned its brain, an entity entirely without the DNA blueprints required to build new replacement parts or direct new physiological functions, truly be considered living? Are those red blood cells actually alive, or have they crossed a threshold into becoming highly complex, slowly decaying biological machines, simply running out the clock until they are recycled? Think about that philosophical paradox the next time you consider the life flowing through your own veins.
That is precisely the kind of rigorous, boundary-pushing question that reductionist biology demands we ask. It forces us to examine where chemistry ends and biology begins. Keep questioning those boundaries, keep visualizing the physics behind the structures, and keep exploring the microscopic metropolises operating relentlessly inside you.